Hello friends, welcome to Sethi Botany classes. In today's lecture, we'll continue with the tissues. In the last lecture, we discussed about different types of tissues like simple tissues, complex tissues. In this lecture, we'll discuss about simple tissues, second type, colon-chymatous tissue. Parenchyma we already have discussed. Now, we'll discuss about colon-chymatous tissue. This tissue is again thin walled and the wall of the cells is made up of cellulose and pectin. Now, colon-chyma cells also have thickenings at certain places. So, because of the thickenings at different places, colon-chymatous tissue is divided into three categories. Angular colon-chyma, lamellar colon-chyma, and lacunate colon-chyma. Now, angular colon-chyma is the colon-chyma which has thickenings at the angles. Now, I am going to draw how to make that colon-chyma. This is the diagram of angular colon-chyma. Now, this colon-chyma will have thickening at the angles. Now, with red color, I am showing the thickenings at the angles. Like this, we can make its diagram. Now, this is the angular colon-chyma, now thickening at the angle. Now, next is lamellar colon-chyma. Now, in this case, no thickenings will be present. Lamellate type or plate-like thickenings are present like this. So, these are the thickening like plate-like. So, red color is showing the thickening material. So, these are plate-like or lamellar thickenings. So, this is called as lamellar colon-chyma. Now, thickenings when present at the angles, angular colon-chyma. When they are tangentially present, thickenings are present tangentially along the cell walls. This is called as, now this is wall thickening, tangentially present along the cells. So, this is called as lamellar colon-chyma. Next, we will discuss about lacunate colon-chyma. Now, here thickenings will be present along the lacuna or intercellular spaces. Now, this is the intercellular space. Now, along this intercellular space, we will find the thickenings. I'll just enlarge it to make you understand. Like this is one cell, this is another cell, this is another cell, Now this is the intercellular space. Now thickening will be born at this lacuna like this. So this is lacunate collenchyma. So we have three kinds of collenchymatous tissue: angular, then lamellate, and third is lacunate collenchyma. Its cells are not completely thickened, they are not completely thin. So thickening is deposited at different places. So depending upon thickening, we have categorized into three categories. Now what are the functions of the collenchymatous tissue? When like parenchyma is completely thin walled, collenchyma is having thickening at certain places. So this will give mechanical support to the tissue. Like this usually collenchymatous tissue is present below the epidermis tissue. If we see the anatomy of the tissues, now collenchyma will be present, whether it is lacunate, angular or any kind. Now this is present below the epidermis, which is also called as hypodermis tissue. Now this is, I am going to showing you how collenchyma tissue is present in the hypodermal tissue. Now this is the hypodermis below the epidermis. So this collenchyma becomes part of the hypodermis in the stem or in the case of roots. So this is present below the epidermis, and this also provides the mechanical support. And collenchymatous tissue, when it is having chloroplasts, this is called as chlorenchymatous tissue, and this will help in photosynthesis. When it has chloroplast, so this will do photosynthesis, make food for the plant. Collenchymatous tissue is usually present in the primary structure or growth of the plant. Now when secondary growth takes place, and this collenchyma cells, again I will show you how this occurs. Now this is the epidermal tissue. Now below it is present, like we have discussed already. You can see this we have discussed. This is the collenchymatous tissue. Now this collenchymatous tissue can again regain the power of division. This can form cork cambium, very important. Now collenchyma cells will again turn meristematic in nature. So here you will see that a layer will get differentiated in the collenchyma only, and collenchyma develops from the cork cambium develops from the collenchymatous tissue. Now this is cork cambium, which is developing from the collenchymatous tissue, and this is also regaining the power of cell division, turning into meristematic tissue, and this will help in formation of periderm. This will cut off cork toward the outer side and secondary cortex toward inner side. Like this will cut off secondary cortex toward inner side and cork tissue toward the outer side. This will be called as periderm, and this development is taking place here in the collenchymatous tissue. So collenchyma also help in formation of the cork cambium and also help in development of the cork tissue during the secondary growth of the plant. This provides elasticity to the plant, this provides support to the plant, and when this bear chloroplast, this will help in photosynthesis, and when it stores reserve food material, this may store reserve food material. So, this will store starch grain, protein or oil crystals in the colon cametus tissue, and whenever plant needed those substances as nutrients, they will be provided to the plants. Thank you for watching my video.